Good morning, everyone. My name is Godae Nwineewii. I'm a rising senior at Chippewa Valley High School. And this past summer, I worked under Dr. Moore's lab under Grace Swanson, who was my mentor. And my project was finding the role of benzene in ACOD1 expression. So in order for us to understand, we need a little background into what exactly we're talking about. So we'll start with air pollution. Air pollution occurs when harmful gases and dust are released into the atmosphere. 90% of people breathe in polluted air daily, and around approximately 7 to 10 million people worldwide die each year. Air pollution can actually contribute to the spread of diseases. In highly dense air polluted cities, they have more diseases than less cities that have air pollution. Common gases include carbon dioxide, sulfur dioxide, ozone, lead, and what we're studying, which is benzene. Now, the causes of air pollution include industry and energy supply, dust, household energy, and transport, to name a few. Now, the average human actually breathes around 20 million parts per million, 2.5 particles a day. Now, the actual health harms of air pollution can range from skin aging, which can actually lead to skin cancer, strokes, heart attacks, insulin resistance. But actually, what we're focusing on is premature birth and low birth weight. Now, benzene is a colorless or light yellow chemical, which is liquid at room temperature and is widely used in the industry in the United States. It's produced in the environment from car emissions, forest fires, gasoline, and etc. It causes cells in the body not to function properly by activating inflammation. And when that occurs, cancer can start to happen. Now, benzene is actually metabolized in the liver. And then from the liver, it forms all these different types of metabolites, such as benzene oxide, phenol, hydroquinone, and benzoquinones. And exposure during pregnancy is known to affect the development of the child. We're using hydroquinone not only because it's safe for us to use it in the lab, but also we've seen a lot of pregnant women that have used hydroquinone because it's found in skin whitening medications because there's a condition called pregnancy melasma, which happens due to the hormonal changes in pregnancy, which causes brown spots to occur on the face. And a lot of pregnant women use this. So if we're going to use any surrogate of benzene to test, this one's one of the best to use. Now, our gene of interest is ACOD1, which is also known as immune responsive gene 1. It's a regulator of immunometabolism and inflammation and infection. Normal ACOD1 expression may limit pathogen infection and promote embryo implantation, which is why we're looking at it. And this is the pathway for ACOD1. So basically, cis-aconitase stimulates the production of a taconate, and between that, ACOD1 is produced. And then actaconate stimulates and creates mesaconate. And when that is stimulated, this inhibits things such as ROS, glycolysis, but what we're mainly focused on is inflammation. But also, when antagonate is produced, it acts as a negative feedback loop, and it turns off SDH and one of the entries into the citric acid cycle. Now, what we're going to be looking at is going to be in the placenta, which provides the baby with oxygen, nutrients, and antibodies. Also removes carbon dioxide and harmful waste from the baby, and it produces hormones that helps the babies grow. And the main component of the placenta are trophoblast cells, which provide nutrients to the developing embryo. And there's many types of trophoblasts that differentiate from the blastocyst, which is this right here, further down in pregnancy. The cells we're working with are called 171 cells, which are extravilous trophoblasts. And extravilous trophoblasts invade deep into the uterus wall and function to establish critical tissue connection in the developing placenta, which is important for us to see if we're looking at the effects of how benzene affects the growth in the placenta. In order to, what we're using is, which is a transcription factor, which is called twist knockout, and responds to inflammation, interferon beta signaling, which results in gene expression controlled by twist. So ACOD1 is stimulated by interferon beta signaling, which means if twist is knocked out, then there will be potentially more ACOD1 expression. And this is the twist one pathway. And why we used ACOD1 was because the Moore lab did RNA sequencing and found that There's significantly more ACOD1 in male control in males and females than the one without benzene exposure when exposed to 50 ppm of benzene. Now, the main objective is to find how benzene affects ACOD1 expression in the trophoblast cells, which are also known as Swanson 1 cells in the placenta. Because ACOD1, we're seeing it should increase during inflammation, but we're seeing a decrease in it. And it may be due to the signals that occur in the trophoblast cells rather than the immune cells or macrophages. Now, the methods we used was first that we treat the cells with either 15 micromolar or 25 micromolar of hydroquinone. We either let them incubate for 4 or 8 hours, depending on what data points we need to see. Then after that treatment was done, we then collected and colored up the cells in order to form RNA extraction from them. Once RNA extraction occurred, then we made DNA synthesis from it, and then we did qPCR, and then we interpreted the results in R. So our results for that, we saw for the wild types relatively have the same, but when we look at the knockout cells, there's especially a huge difference between the wild types, especially the knockout 25 cells. And for the 8-hour response, it's similar. The wild types here, they're relatively the same, but when we look at the knockouts, we can see higher levels. So our conclusion is that when exposed to the hydroquinone, TWIS1 must be removed in order for ACOD1 expression to increase. And longer time points may be needed in order for us to see how ACOD1 can express. Our future directions are more experiments with different dosages and times to further extend the hypothesis. And I'd like to thank everyone that's listed on the side here.